Yogah Karmasu Kaushalam.
Hello everyone, I'm Hetal Roy from Department of Zoology, the MS University of Baroda. Today I'm going to talk about epigenetic, gene on and off. The human body is made up by so many cells. We have a neurons, we have a pancreas, we have a bone cell. When we have a neuron, we know it is involved in the conductions of the impulse. And when we have a pancreas, the beta cell of it is involved in a synthesis of insulin. Both the cell has a 2N DNA. Why? The beta cell is involved in a synthesis of the insulin and why the neuron is involved in a conduction of the impulse. Furthermore, when we observe the human, we can easily identify they belongs to which continent. We can identify Asians, we can identify the South African Negroid one. How? All the human has a 46 number of the chromosome, or we can say 23 pairs of the chromosome. How the humans are different from each other. That is something is there which give us identity as continent specific, habitat specific, regional specific we can say, or the each cell has a, some kind of the pattern. Okay, with that it can understand what to synthesize, what to do, and what not. So there is something beyond the genetic that is known as the epigenetic. It is emerged in the last few decades, which studies the factor that influence the gene function, not the changing in a gene sequence, but how the gene changes function on the basis of the tissue type, cell type. The term epigenetic was suggested by the developmental biologist Wannington in 1942. So this is not a new word. So, what is epigenetic? That means without the change in a sequence, there is nothing like a recessive one, nothing like a dominant one. Okay, there is no mutation in that. Only there is a modification occur in a nucleotide, or we can say the amino acid, and it regulate the gene expression. That we say the epigenetic. So the epigenetic, that means the study of mitotically and or meiotically heritable changes in a gene function that cannot be explained by the changes in a DNA sequence. So epigenetic refers to the stable heritable trait not explained by changes in a DNA sequence. It is a Greek word and it suggests that on top of genetics. The chromosome modifications that affect gene expression, that means by the modification of the histone or the DNA methylation. You can see over here, this is a DNA methylation over here and the histone modification. The essential for the normal development. So during the implantations, okay, something govern which gives the identity to the embryo and the methylation, the de novo methylations occur that it governs the development of the embryo. It can modified by the environment. When you are continuously in exposure of one or two chemicals, you are living at the polluted area, definitely the epigenetic pattern it will be changed and it can be disrupted in disease condition. So the types of epigenetic modification, it is a DNA methylation where the nucleotide or you can say the nitrogen base is methylated. Second, it's about the histone modification, histone methylations, histone acetylation, histone phosphorylation, uh, ubiquitinations, ribosylations, sumosylations. So these are the what we can say the histone modification. And furthermore, okay, the chromatin modeling with the help of the bromodomain or the chromodomains of the nucleosome, where the acetylation and the methylation are the regions of the nucleosome. Furthermore, one more modification, it is RNA silencing. So the non-coding RNA are involved in the regulations of the post-transcription. So when we have this with the three main mechanism, okay, which governs the epigenetic, that is a histone modification, Okay, you can see over here at the level of the nucleosomes, the different histone proteins are involved in it. Then after the RNA interference, or we can say the RNA silencing. So here the two main non-coding RNA, one it is a microRNA and second it is a short interfering RNA. Generally we say it is siRNA, which degrade the mRNA. Okay, and there is no further translation after that. So it is a post-transcriptional regulation, and then after the nitrogen base, okay, which it gains the methyl group and the DNA methylation. So these are the three known mechanisms of the epigenetics. So we talk about the DNA methylation. It is a process by which the methyl group are added to the DNA molecule. The DNA molecule that it is made up by the four nitrogen based, that is adenine, cytosine, guanine, and thymine. So here the cytosine at the 5' positions of the cytosine, there is adding of methyl group with the help of the enzyme, that is a DNA methyl transferase. Generally it is known as a DNMT. So DNMT1, when we talk about it, is the regulator of the DNA methylation, which is the you inherited, and whenever there is a de novo pattern is there, the new methylation pattern you gain. Okay, so that is a DNMT3A and the 3B. These are the two enzymes, okay, which are involved in a de novo synthesis of the methylation when you gain the new pattern of the methylation from the your right methylation pattern or the inborn methylation pattern that you have. It is so when we have the methylation, the cytosine, okay, it turns into methylated. So we can see over here, this is the cytosine, and with the methyl transferase, okay, the cytosine gain the methyl group, okay, and it turns into the 5-methyl cytosine. This 5-methyl cytosine is now methylated, and this its form, okay, that is the CpG island. So the CpG island, how the CpG island is formed. So here first of all we see about, okay, this is the as adenosine methionine. This adenosine methionine, it is the part of the 1-carbon metabolism where the methyl group is transferred. So this adenosine methionine transfer the methyl group into the cysteine, okay, at the 5' position, and then it turns into the as adenosine homocysteine. Now the cysteine, okay, it form here, we can see it is a CpG island. So this is a CpG island. So here when the cytosine is methylated, it has it is linked with the guanine with the phosphodiester bond. So here this C will be a methylated, and this region, okay, this is not the cursor is moving, okay, this is not a pattern, we can say the CpG island. This is a CpG island when the C is the link with the G with the phosphodiester bond. Hydrogen bonds is not involved to formation of the CpG one. So here you can say where the CpG islands are present in our eukaryotic genome. So CpG islands are present at the 5' untranslated region, 5' UTR. The 70% of the CpG islands are present at the 5' UTR region. Okay, that is the chance this CpG island is present at the coding region of the gene 2. So whenever we are targeting about the methylation pattern, to study about the methylation patterns of the gene, we always target, okay, the 5' UTR region. After that, we have the histone modification as an epigenetic marker. So we know what is a histone. So the histone, it forms the nucleosome, the octamer, okay, which is made up by the 4 histone, that is H2A, H2B, H3, H4, and the one linker, the H1, which is most common, and then after the H5. So this our DNA is wrapped around this octamer, the histone protein, or we can say the nucleosome. So when this DNA, it's wrapped around the nucleosome, okay, and the histone, there is no modifications, okay, then this DNA is tightly packed with the nucleosome, and it is a condensed, and it never allow the transcription factor, okay, to set on it, and there is no transcription. So it is a regulations, okay. So when the transcription is required, this chromatid has to be open and allow the transcription factor to bind to it, and the transcription has to start. So what happened at this time? This histone, it's regulate the DNA packaging with the immense influence on the degree of the chromatin compaction. So it is a condensed chromatin influencing on the transcription activity as well as the transcriptional silencing. What are the modification of it? So the modification of histone include the acetylation of the lysine residue. The lysine is symbolized with the alphabet K. So the acetylation of the lysine residue, methylation of the lysine and the arginine residue, phosphorylation of the serine and threonine that we know very well, and the ubiquitination of the lysine residue present on the histone tail, as well as the sumoylations and ADP ribosylation. So these are the patterns of the histone modification. 10 by 10 we'll see each one. So the histone acetylation is carried out by the enzyme. It is called the histone acetyltransferase HAT, that are responsible for adding the acetyl group to the lysine residue on histone tail, while the histone deacetylation, that is known as the HDAC, are those that remove the acetyl group from the acetylated lysine. Generally, the presence of acetylated lysine on histone tail leads to a relaxed chromatin state that promotes transcriptional activation of selected gene. In contrast, deacetylation of the lysine residues leads to the chromatin compaction and the transcription inactivation. Now, what happened? When the lysine is deacetylated, the lysine is a positively charged and it binds to the negatively charged to the DNA and it makes the condensed structure. When the lysine is acetylated, it loses the positive charge. Now, when it loses a positively charge, the negative repulsion occur and chromatin become open and serve for the transcription factor to bind to it and promote the transcription. So this is the histone acetylation. So here we can see, this is a closed chromatin, where the histone is deacetylated. When the histone acetylation occurs, the chromatid come into the relaxation state and it is the open configuration. So when it is open, the transcription factor, it is easily invaded by the transcription factor and the transcription occur. So the gene switch on and the gene expression start. So here we can see this is a lysine with the positively charged with the N-terminal and so binds with the DNA molecules and compact. It gives the compactness. But when it is the acetylated, with the negative or it loss is a positive charge, so that the repulsion and it cannot form the compact structure and the compactations occur and the condensations occur and then the chromatin it's open and ready to bind with the transcription factor. So here the deacetylated histone is associated mostly with the heterochromatid, so the compact chromatid, and then after the acylated histones are associated with the euchromatid. We can see over here it is not that compact and it's it is an open structure of the chromatid where it is allow the transcription factor to bind it and the further the cascade, that is the gene expression start. Here after that we have a histone methylation. So the histone methylation, it may be monomethylation, it may be dimethylation, and it may be trimethylation at the N-terminal end of the H3 histone. So the arginine is involved in a methylation. The arginine residue it turn into the methylated form, or we can say the modifications occur at the arginine residue. So the H3 histone, when it is a heterochromatid at the N-terminal end of the H3 histone and the 9 number of the arginine and 27 numbers of the arginine, that is the trimethylations occur and it remain a compact. But when we have a euchromatid, at the H3 histone itself, the 4th number of the arginine, where the 2 additions of the methionine, that is a dimethylation or the trimethylations occur. So here we can say this is acetyl 1 group of it, and here it is a heterochromatid which is a highly compact. So when it is a highly compact, it is not allowed, that is the open chromatin structure, and we can say suppress the expression, turn off of the gene expression occur. But when it is with the residue of this, okay, K4 is involved, that is the arginine on the fourth position, it is involved at the N-terminal and, okay, that is the open chromatid, and then after it allows the expression to start. So then after we have a phosphorylation modification. So here the phosphorylation and the N-terminal and of the histone, there is a serine and the threonine amino acid. The serine and threonine, we know that they are the phosphorylation. So the serine and the threonine are phosphorylated. When the serine and threonine is phosphorylated, they are the negatively charged, and when they are negatively charged, there is a repulsion with the DNA molecules, which is wrapped around the nucleosome. And when the repulsion occurs, the chromatid, it becomes open structure. Now the chromatid is active, or it is, we can say, it is ready, okay, as a switch on for the gene expression, and the transcription begin. Then after the one more modification, that is the ADP ribosylation. So when the ADP ribosylation, so here when the ADP ribosylation imparts a negative charge to its modified residue, and when it is a negatively charged, again the DNA it's repel, and when the DNA is repel, okay, it's turned into the open configuration. So when it turn into the open configuration, it is ready for the gene expression. Now it allows the translation factor to invert the region where to set and and the transcriptions occur. So when the ADP ribosylation is there, it is specific with the heat shock protein. So the nucleosome which are involved with the heat shock protein region of the gene sequence, okay. So at that nucleosome, the ADP ribosylations occur. So whenever there is the inflammation signals or the infection signal, the heat shock protein, it has to be activated, okay, as a defense reaction. And the nucleosome involved with the heat shock protein sequence, okay, where the heat shock protein gene is wrapped down the nucleosome, okay, where the that is ADP ribosylations occur, it come into the open configurations, and the allow the gene expression of the HSP gene. Okay, so this is about the ADP ribosylation. Now then after this something we can we can say it is a remodeling. So that is the some bromodomain and the chromodomain and the remodeling of it. So when the bromodomain protein is there, it's bound to the acetylated region, and the chromodomain is there, it's bound to, that protein is bound to the methylated region. The furthermore, the bromodomain chromodomain complex which forms, so that the complex, the one side it is bound to the acetylated and one side to the methylated. This pattern is identified, okay, and then after the transcription factor is recognized, okay, where to switch on, where and where not to the switch on, okay. So this we can say as a barcoding pattern, okay. So each region of the histone where the specific gene is wrapped around, that is a pattern by masking by the bromodomain and the acetylated and the di and the trimethylated by the chromodomain, and it forms the barcoding pattern. On the basis of the signaling, our cell recognize this barcoding pattern and the gene expression occur. The last modification, it is about the RNA silencing. So we have a non-coding and the coding RNA. mRNA, we say it is a coding one. So at the end of the transcription, we have a mRNA, and then after, when that is the some a kind of the signal where the and that mRNA need not be transcribed, it is regulated by the non-coding RNA. So the two main non-coding RNA, that is a microRNA, and the second, it is a small interfering RNA, siRNA. So this siRNA identify and they bound to the microRNA and they bound to the mRNA, from the dimerization circle, two strands, double strand formation occur, and then after the that post transcriptional regulation and the mRNA will be degraded. Now, what are the effect of the epigenetic modification? So, the effect of the epigenetic modification, that is, it is the regulate the transcription. Where there is a requirement of the transcription, the gene become expressive, and where there is no requirement, the gene remains silent. So, wherever there is a change, like we can say when the methylation is there, it is a That time the transcription factor cannot bind with the promoter region and the gene remains silent. When it becomes demethylated and it becomes hypomethylated, the transcription factor recognize it can set on that promoter region and the transcription start. So there's a switch on, and the switch on the basis of the methylated pattern on the DNA. So this is the identification of it. So in the when we talk about the X chromosome inactivation, okay, one of the X chromosome inactivated, which is highly methylated, and or we can say the doses compensation, that X chromosome it will not remain expressive. Okay, so this is the we can say the modification, okay, of the epigenetic, and we can observe that, okay, in some of that it remain methylated, and that's why in the particular tissue or the particular cell type, okay, it will not remain expressive. Okay, it is furthermore I can say the barcode pattern is there, it cannot okay, read by our cell, and that cell will not okay, react on the basis of the signal. The factor that can influence the epigenetic. So that when we say the epigenetic, it's not always remain imprinting. There is a chance, okay, it's changed. So the few of the marker, okay, are there few that it's changed the epigenome. So one of that you can say about the pollutions, medications, infections, smoking, alcohol, nutritions, toxin, okay, these are the thing, okay, it affect the epigenetic pattern or the imprinting of the epigenetic. So when the person, just we take the example about our lifestyle, we are taking the junk food when the micronutrient and the macronutrient is not a balance. So when the junk food, when the some of the vitamins like we can say the B12 and the B9, that folate, okay, is deficiency you have. So the folate, it's an important role in a one carbon metabolism, and it is in a one carbon metabolism. We know that is a synthesis of the as adenosine methionine occur. This as adenosine methionine is a donor, okay, for the methionine for the donor of the this methyl group to the cysteine. If the folate deficiency is there, there is no as adenosine methionine in the ample number, okay, and then the that cytosine is not a methylated and it remain hypomethylated. And when it is a hypomethylated, when the gene remain need to be silent, it cannot be remain a silent because it is hypomethylated and continuous. Okay, there is a synthesis of the mRNA occur, and it's create a problem. Okay, it may trigger some kind of the pathogenic condition. So this is we can just simple example about the nutritions. It's okay, and other factors are there. The very common factor when we have a habit to go to bed at the late night. Also, it is also one of the uh, we can say the factor which affect the epigenome. Other than that, okay, you can change your epigenetic to the good aspect that you can say about the exercise, okay, the healthy diet. These are the good practice, okay, and you can imprinting, you can gain the imprinting, okay, and you can overcome from that the bad epigenetic printing, okay, that happen because of the pollutions and the toxin and all. There are the many diseases are associated with the when the epigenetic pattern is changed. Okay, the one of that it is a cancer. So we know that there are the gene which is a tumor suppressor gene and the oncogene and the proto-oncogene. So when we say about the tumor suppressor gene, when the tumor suppressor gene, whenever there is the some kind of the change is there, the tumor suppressor gene, okay, the expressions occur and it regulate the expression of the uh, oncogenic gene. But when this tumor suppressor gene, it is a hypermethylated, and the oncogene, it is a hypomethylated. Okay, so when it is a hypomethylated, that means continuous expressions of the oncogenic mRNA, and then after we have a translation product that is a protein, and when it is a hypermethylated of the tumor suppressor gene which control the functions, okay, of the oncogene, or it control the expression of the oncogene, that cannot be happen into the cell. Okay, so this is only the change in a methylation pattern in a cell. Okay, the person, it's suffering from the cancer. So here are some selection of the 20 gene, okay, of the healthy gene. The dots are related to the methylation status of the gene. So when it is orange, it is a methylated, and when it is a dark color, it is the unmethylated. Okay, so the unmethylated, the remaining gene, it is unmethylated, so that's turn into the switch on, and when it is methylated, it turns into the switch off. So these are the 30, and when we study about the cancer progression, we can see over here, okay, the some of the example over here, that is the 5, 7 and 8, okay, it turns into the methylated stand, okay, which previously in that they were not methylated. So when that is turned from the methylation to the demethylation, hypermethylation to the hypomethylation to the hypermethylation, okay, it changed the expression of the oncogenic gene and as well as the tumor suppressor gene and involved in a cancer progression. So here you can see about okay, the experiment which suggests how the cancer progressions occur with the study of the methylated and unmethylated gene. Here about the epigenetic and cancer. So by understanding more about the methylation of the particular gene, we can better understand the pattern in a cancer progression, which can help to inform the prognosis and the treatment. If we identified, okay, in a particular lineage of the cancer, what kind of the genes are become hypomethylated or the hypermethylated, we can make a treatment strategy or we can make a diagnostic tool, okay, and we can okay, treat the patient in a better way, or we can form the treatment regimens. So this is about the epigenetic pattern, okay, with that related to the cancer, but we can explore this epigenetic pattern in other disease and understand the pattern of the, we can say, about the disease. Furthermore, we can say we can use this epigenetic in a development too. So during the embryogenesis also, the cell get its fate, and on the basis of that, okay, they identified what to be. So the neuronal cells become a, okay, we know that it is the from the ectoderm, okay, the central nervous system form or the neural tube formation occur. If there is a change in a methylation pattern, okay, gene that need to be expressed you during the formation of the neural tube, okay, if they remain silent, there is neural tube defect, okay, occur, or we can say there's some kind of the neurocrystallopathy that is related to the developmental deformity. So we can use the this epigenetic pattern, okay, that the normal cell uh, perform during the embryogenesis, okay, according to the fate of the cell, and when there is a disease condition or we can say the abnormal development. So this is the epigenetic and the disease, that is one of that which is known as a Prader-Willi syndrome. So in a Prader-Willi syndrome, in a maternal copy, okay, it's remain the switch off, okay, imprinted like that, and in a paternal copy, the gene remain active and remain switch on. But when it is a Prader-Willi syndrome, here we can see in a maternal copy they remain switched off. But in a paternal copy, the genes are defective and they cannot remain expressive. And in that, okay, if there is a change, okay, about the DNA methylation pattern, gene remain inactive, that turns into the syndrome, that is a Prader-Willi. So in near, okay, the previous year data, when we talk about the chronic disease and the epigenetic pattern, here the number of the PubMed publication, you can see on the epigenetic publication, okay, increasing up to the 43%, okay, and the chronic diseases, okay. So the in chronic diseases also we implicate about the how to identify, okay, the epigenetic pattern, okay, change in epigenetic pattern and impair the normal physiology and leads towards the disease condition. There are some of the mechanism with the help of that we can study about the epigenetic pattern. So when we wish to study about the DNA methylation pattern, methylation study, we need to convert the DNA into the bisulfide. Okay, so the bisulfide conversion, it is a common method. Okay, when we need to study about the methylation pattern of the DNA. So what we do, there is the bisulfide conversion. So when we isolate the DNA, we keep this DNA into the bisulfide conversions reagent. So at that time, when the cytosine, which is a methylated, it remain as a methylated, but the cytosine which is not methylated, okay, which is unmethylated in a DNA, that cytosine turns into the uracil. So when it turns into the uracil, you can identified, okay, which region, okay, where the methylation is there and when it is the unmethylated. So when there is a change in a imprinting, epigenetic imprinting, we can get the idea. So after the conversing, the bicyclic conversation, we can use the DNA for the next generation sequencing. We can do pyrosequencing, and if we don't want to go for the sequencing, we can do the qPCR study. So with the help of the myth primer, we design the specific primer when we are studying about the qPCR, okay, about the methylation pattern or the unmethylation pattern with reference to the disease conditions, or we can say about the developmental condition. uh, then after, you can do the chip-seq, okay. So that is about the related with the histone. So those histone are histone where they your specific gene of interest, okay, wrapped around the histone. So we can do the chip-seq, okay. So the chip-seq gives the data of the DNA protein interaction. So those DNA wrapped around the histone and the DNA sequence that you want, or the DNA that you want to specific gene specific one, okay, you can study with the help of the chip-seq, and you can validate further with the help of the qPCR. The chip-seq, the kit is available. We use that kit, okay, we can study about this pattern. And about the ELISA assay, we can do about the acetylation pattern. So the histone acetylation and deacetylation, the ELISA kit is available. Okay, simply just we can come to know about the histone acetylation and the deacetylation with the help of it. Whenever there is a modification occur, there is a change in a chromatin configuration. So there is open structure of the chromatin configuration or the closed structure. Simply with the help of the host stain, okay, or the DAPI, we can understand about the chromatin configurations with the help of the fluorescence microscope when you are using the DAPI. Furthermore, when we want to study about the chromatin configuration, there is 3C model is available, okay, and with the help of that, we can study about the chromatin configuration. Uh, there is a browser for the epigenetic data. So here there is the list of the browser. Epigenetic database. There is again here is the list of it, and the bisulfide primer design. So when you wish to study about the DNA methylation pattern, okay, you need to design the primer. So you can the MyLab is using the myth primer. Okay, we design for the sequencing also with the myth primer, and when we validate with the qPCR, you can design both the type of the primer with the help of the myth primer. Um, uh, Many a time, okay, when you want to just specify the specific sequence of the 5 prime UTR region, okay, we can do the Sanger sequencing. So you identify the 5 prime UTR region, and up to the 2000 base pair sequence, okay, you will get the enough CPG island, like 6 to 7 CPG island up to the 2000 base pair. Okay, we can go for the Sanger sequencing of our defined uh, coding region, or the define, or you can say the desired gene of interest. So that also we can do the Sanger sequencing of the that the 5 prime UTR region, when you specify the region that with the minimum number of the 7 CPG island. And when you want to do the methylome study, you can go to the sequencing. So this is and for the presentation. Thank you all.